Okay, hello and welcome to the ZOOMinar. Today's speaker is Marina Ramirez-Alvarado, professor from Mayo Clinic, and we have also guests for the panel, Dr. Ben Drive from Munich. He will be hosting the presentation. He will introduce the speaker and moderate the Q&A session. Ben, take over.
Okay, it's my great pleasure to introduce Marina Ramirez-Alvarado for the ZOOMinar today. Actually, Marina originally is from Mexico, but we have a connection because she did her PhD in Germany, so at the EMBL in Heidelberg in the group of Luis Serrano and Francisco Blanco. And there she worked more like on biophysics and thought about the formation, stability of beta hairpins beta hairpin structures in polypeptides. And then she slowly went in direction of amyloids, where she did a postdoc in the group of Dr. Lynn Regan at Yale University, where she looked at the influence of protein stability on amyloid formation before she actually went to Minnesota, to Rochester, to the Mayo clinics, where she is since around 2002, if I'm not wrong. And she was there since then, a pioneer in the study of systemic amyloidosis to find out what actually makes a protein, a light chain protein aggregate, which factors influence this. So she looked at the role of mutations in specific VL structural motifs that maybe affect the aggregation propensity. Then she looked at the conformation around the dimer interface. She coined the word alternate dimer with respect to canonical dimers, and looked at the stability and the influence of this particular dimer interface on the aggregation propensity. She also studied partially folded intermediate states and characterized their role for amyloid formation. And the title of the day is When Antibodies Misbehave, the biophysics of aggregation, toxicity, and rescue in vitro and in cell culture. And I'm looking very much forward for your presentation, Marina. So please go ahead. The stage is yours.
Thank you, Bernd, and thank you, Rams, for the kind invitation. So today I am going to, let me put my mouse in the right place. Here we go. So I want to give some overview of the topic for those of you who are not familiar. So we're going to review the immunoglobulin domain structure and where the structure diversity comes from. And I encourage all of you to write your questions as things go on. And I hope to allow enough time for questions. I'm going to give a disease overview from the clinical perspectives because a lot of you are probably not very familiar with the disease and the complexity associated with it. Then I'm going to give a brief overview of the previous biophysical data that we have published from the lab, particularly the bacterially expressed variable domain protein that Bernd was referring to. And I am going to give you a comparison between the bacterially expressed and human cell expressed full-length light chains. This is the newest data that we have in the laboratory. It's so new that I was not even able to include a graph that my summer students sent me this morning because I needed to take a shower. Too much information, my son would say. I want to link the biophysical information that we have, the structural information that we have, to then the studies that we have done with these same proteins doing cell internalization toxicity associated with these proteins, and then some studies that we have done about the rescue of these light chain-based toxicity. And I know that I mentioned aggregation in the title, but I want to concentrate on the earlier part of the talk, mostly because that's the unpublished data. But if we have time, and I'll make time to at least show you the highlights of the very extensive biophysics and kinetics of amyloid studies that we have been doing for the past couple of years, just to show you a little bit of the detailed information that we can derive from analyzing these early events of fiber formation. All right, so here we have the immunoglobulin structure, and let me, I hope you can see the highlighter, and if you cannot, one of the moderators can let me know. So the immunoglobulin, in this case, I'm showing you the IgG, has a heavy chain in red, and these are the numerous domains, and then the light chain in yellow. And the light chain has two domains, the variable domain, which we have here further highlighted and zoomed in, and in green, we are depicting the framework. This is the structural part of the domain, and it is an immunoglobulin fold. It's a Greek key domain, and it is composed mostly of antiparallel beta sheets, although this N and C terminus, it's the only region where it's a parallel beta sheet. The pink regions that I am highlighting here correspond to the loops in the protein that bind the antigen. This is the business end of the protein. So in a regular antibody, these ends between the heavy chain variable domain and the light chain variable domain, they bind the antigen. And these loops have very funny names called complementarity determining regions, or CDRs. Very important information. I'm going to be talking about this quite a bit. There are 73 light chain variable genes in humans. 40 are kappa and 33 are lambda. So there are many different genes that the immune system can choose to make these proteins. And this process starts with gene rearrangements. In this image, I'm showing you the kappa decision-making in the gene rearrangement. So there are 40 variable genes, and structurally, the variable domain is composed of the V gene, the variable gene, and the junction gene. The last strand corresponds to the J gene. So this is called the germline DNA, and that's why you will hear me and many people in papers call, oh, which germline does this protein correspond to? So from these 40, the B cells decide at certain stages of differentiation which variable domain is going to be chosen. And then it's going to choose one of the J segments. In this case, there's 4 to 6 J segments. So that decision is made. In the kappa example, there's only one constant domain. So that is not a decision. It is done. And you come with a functional gene. The most frequently subtypes in light chain amyloidosis are 5. There's only one kappa, the kappa 1. And there are 4 different lambdas that are very overrepresented in light chain amyloidosis. And this is going to be very important because I'm going to be telling you a lot about kappa 1 proteins that we have studied in the laboratory. And there's going to be a very notorious lambda 6 protein we're going to talk about. Furthermore, so you selected this gene out of the 73 genes, but then you're going to add more sequence variability by the process of somatic hypermutation. And this is a graph that shows, based on the residue number of the variable gene, the variability that you find through the process of somatic hypermutation. Not surprisingly, these loops that are responsible for antigen binding from these antibodies accumulate more mutations, especially CDR3. But the framework regions also have some mutations. As a result, every light chain, from the 1 million or so that each one of you 1 million different antibodies, immunoglobulins that have light chains, variable genes. Every single light chain has a unique combination of somatic hypermutations. This is a result of the germline genes, the B, J, and C genes that were chosen, plus the somatic hypermutations. We've done a lot of sequencing. I'm not going to talk about that, but there has never been a wild type or identical germline sequence that we have seen in the sequences that we've done, either in normal repertoire or pathogenic light chains in our studies. Now, let me introduce you to the disease. So in your normal generation of immunoglobulins, let's say you got a summer cold, hopefully you haven't gotten COVID, but some of you probably have. Your bone marrow, it's where B cell differentiation happens. And the most differentiated B cell is called a plasma cell. Plasma cells are responsible for secreting antibodies. And a plasma cell is a protein factory. They are secreting antibodies, especially during an infection stage. But in the case of light chain amyloidosis, there is an abnormal proliferation of monoclonal plasma cells. What do I mean by monoclonal? Monoclonal is when you have one clone, you are making only one type of antibody. And then the secretion is also defective or altered because you're either making an excess of light chains compared to your immunoglobulins. So there's the two being made, intact or free light chains. And these are the immunoglobulins molecules that we see in light chain amyloidosis, or you are not making complete immunoglobulins at all, and there's just free light chains. These immunoglobulins are in circulation, and in circulation, they go misfold, internalize, aggregate, and deposit in the extracellular space of organs. The most commonly affected, kidney and heart, causing organ failure and death. So these are some clinical features in light chain amyloidosis, and we call it AL as a abbreviation. So you're gonna see in my slides AL. It is rare and understudied. There are 12 cases per million per year in the United States. These are new cases. The median age of onset is 62 years, and because that abnormal proliferation in the bone marrow is considered a malignancy, this also matches with the age where most people will develop a cancer. But there's a discussion whether or not this is a malignancy or not or cancer malignancy. It's a matter of nomenclature. The median survival is 12 to 40 months, but this also has to do with the staging of the disease, similar to other cancers. Now, cardiac involvement, median survival is less than a year. And I'm gonna show you a graph of some of the staging that our clinical colleagues have come up with. And the stage 3B by Giovanni Palladini, one of our Italian colleagues in Pavia. And I want you to pay attention to this very steep curve of survival. These patients are those that have very advanced disease and die very quickly. And these are the patients that we are not able to help still with all of the advancement and the management that has happened in the last 20 years since I joined the Mayo Clinic and since I have learned so much from my clinical colleagues. We're still unable to help these patients. So this is what helps me get up every day in the morning to try to understand this disease, to try to do something that in the future is going to help get these patients survive more and eventually be able to live longer and be cured. But these patients give me hope, because when I joined Mayo 20 years ago, the patients that survived more than 10 years were less than 5%. Look at this, 10 to 20% of the patients survive more than 10 years. And this is thanks to earlier diagnosing and better management of these patients. Why some patients survive more, some patients survive less, we don't know yet. But because I'm a protein biochemist and protein biophysicist, I'm going to tell you is the protein. Obviously, it's up for debate. Most patients have 2 or more organs involved. And the only available treatments that are approved by FDA and European agencies are chemotherapy and stem cell transplant. The disease is treated like a cancer. There is no treatment for the protein misfolding and the amyloid deposition, although there are clinical trials undergoing at the moment. Now, the approved treatments are very poorly tolerated because these patients have organ dysfunction or failure. And for chemotherapy to work well or be tolerated, you need your organs to work well. So it is very urgent, and it is really important that these clinical trials are well designed and successful. All right, we're gonna switch here. So if you have questions about the previous topics that I cover, write your questions. So I'm gonna define how I call the proteins in our laboratory, because when I label them in our graphs, sometimes I don't have a lot of room, so I use this abbreviation. So this is an immunoglobulin light chain. The variable domain is, the abbreviation is VL, the constant domain is CL, and the full length protein is called FL. Sometimes people call it LC, or when they are urine derived, they are called BJPs or urine derived. And the proteins in our lab were originally called AL because we study proteins from multiple myeloma, which we call MM. And the number was associated originally with the GenBank ID number. All right, this is a summary slide showing you some of the proteins that we have extensively studied in the laboratory. And in the top panel, we have the proteins that have the canonical dimer interface. Bernd Reif made some comment about this in his very kind introduction. And these proteins make a dimer interface that it's the same dimer interface that you see in antibody crystal structures. And then we have AL09. We're going to talk a little bit about AL09. And then ALT05, we're going to talk about that dimer. And this is a very nice review that we were invited to write that has a lot of these background information. If you don't have time, screenshot. I have a lot of references. This is another good slide to screenshot because I wrote this for a grant and I thought this is a really good summary of years and years and years of work. So I'm not gonna describe everything here, but these are some of the proteins that we have studied. And I'm giving you a lot of information here about the gene. I'm using two different nomenclatures. What is the staging of the patient? So AL09 was a stage 4. The patient died in less than a year after diagnosis. What dimer structure we found by crystallography, the dissociation constant, if we did NMR, what states we found, if it was promiscuous or a single state, if we did toxicity, or we have seen dimer, oligomeric species, I'm sorry, and then a whole bunch of references, and then a couple of reviews. So anyway, but let me introduce you to some of our wonderful proteins from very sick patients. So AL09 comes from a cardiac patient diagnosed after a brief multiple myeloma diagnosis. Patient died less than a year after the light chain amyloidosis diagnosis. And this patient is a kappa-1, has 7 mutations, but the important mutations are in the dimer interface. This is showing the crystal structure of the germline protein that we generated artificially because there's no germline protein being secreted or made in anyone. And the positions are shown here where AL09 has substituted the germline amino acid to different amino acids. And these are the dimers. This is the canonical dimer, and this is the altered dimer interface. And the best way that I have to describe it is the canonical dimer is the way you clap normally. The altered dimer interface, 90 degrees rotation, is how the flamenco dancers clap, 90-degree rotation. Even better yet, I have a movie here. Let me see. I don't know if it's because I have the, oh.
Try going to the non-PowerPoint mode and then see if it works.
Okay. Oh, here it is. Okay. Can you see it?
Yes.
Okay. So what I'm showing you here today is 3 different crystal structures and an NMR structure. And let me go back because it's not a very, okay. So we have in yellow the germline crystal structure, in cyan AL09, and in red it's the reciprocal mutation kappa-1 that goes in position 87, that is in the dimer interface, to the amino acid that is found in this patient, histidine. Okay, here we go. So you have altered dimer interface flamenco, and then there's a 180-degree rotation, so to speak, from the original position. One monomer is fixed, the other monomer is moving, and then in this review, we are also showing you that while the monomer appears to be fixed, there are some side chains that are moving. This movie is in this annual review. If you missed it, here we have to keep going because we have a lot of stuff to show you. This graph is also published, but it shows you here a really interesting correlation of stability, ability to form amyloid fibrils, you know, the time that it takes to form fibrils. And let me orient you. If you have a very small delta G of folding, that means that you are very unstable. Let me go back to my laser point. So AL09 is very unstable, and it takes a very short time to form fibrils. Now, the germline, it's stable, but we actually made some mutants that were more stable. And here we're coming from the 2 ends. We have the germline, which is the wild type, and we have the patient mutant. And we are making mutations from the patient to the germline and from the wild type, the germline, to the patient. In this line shows you when we have histidine at residue 87 and tyrosine at residue 87. And that seems to be an important distinction, and telling us that the residues at the dimer interface are critical for the stability and for the ability of these proteins to form amyloid fibrils. Since then, we've learned a lot more, but also we have evaluated a lot of sequences, and it is a manuscript that has been in my computer waiting for me to share with the world via bioRxiv. And my goal is that by September, this manuscript should be there, so stay tuned. And again, if you have questions, because I'm gonna switch gears, put them in the chat. Okay. Full-length immunoglobulin light chain studies. This is not published, so take notes, screenshots. So 7 years ago, Luis Blancas Mejia and Tim Horn in my laboratory purified from bacteria the full-length protein. Why did they start working with the full-length protein if we spend a lot of years working with the variable domain? Well, the variable domain makes the core of the fiber. But when we started doing mass spectrometry data on the amyloid deposits of the patients, we started finding full-length amyloid light chains in the amyloid. And then we started thinking, well, we need to start studying the full-length proteins. And guess what? For thermodynamic analysis, for those of you that do these studies, what we need to have is reversibility. So they were doing a thermal denaturation, and our proteins have a decrease in the signal of ellipticity. So they are monitoring the beta sheet structure. I'm showing you sort of raw data here. And then the protein was unable to refold. And this is not a surprise, because earlier studies from our laboratory and others, and then further studies that we conducted since then, had shown that urine-derived protein and other proteins that we made in bacteria had these characteristics. So what we started doing in collaboration with Matt Otten and Alex Tischer was scan rate dependencies, where we started trying to derive thermodynamic parameters by doing kinetic studies. This paper is beautiful, it's complicated. Sit down with a cup of coffee or your favorite beverage and read it, because it has a lot of information. We're doing this using CD and fluorescence. And what we are able to do is, from an irreversible refolding, we're able to derive true thermodynamic data. People doing calorimetry have been doing this for many years. Now we're doing it with CD and fluorescence. So these are bacterially derived, and we are doing AL09 full length and the germline. And as you can see, there is a scan rate dependency. So the protein, when you let it rest in between temperature changes, between heating, it is behaving. So at 0.5 degrees per minute is going slowly, but at 2.5 degrees per minute, it is going faster. So the protein is behaving in a different way. And that is what gives you a kinetic control in the protein folding reaction. If we were able to go very, very slowly, 0.001 degrees per minute, we would do it, but then our students will take decades to do, or more, to do their PhDs, and that's not feasible. Now, this is a money figure, people. We started doing our human-derived expression system for another reason. Ask me if you're curious. And we were surprised when we saw that when we express our proteins in human cells, the protein refolds reversibly. This is by CD. This is by fluorescence. And by fluorescence followed in different ways, by the intensity of 360, unfolding by lambda max. And we were puzzled. And we have changed plasmids. We're doing this with a histag, without a histag. We are now removing some cloning artifacts. It's exactly the same. These proteins from humans have the disulfide bond, and it's something that I failed to tell you when I was showing you the light chains, but I'm gonna tell you now. These light chains, they attach themselves to either the heavy chain or to themselves by a C-terminus disulfide bond. And when they have the disulfide bond, if you see thioflavin T intensity here in green, which is a measure to form amyloid fibrils, they don't form fibrils after 800 hours. And trust me, this is a long time. When you follow the disappearance of the monomer, you see some disappearance after a long time, but the species that you see by transmission electron microscopy are actually not fibrils. This is dynamic light scattering, and these species are monomer and dimers that then start forming something that are, you know, some oligomeric species, but nothing really that would explain a fibril. And by CD spectroscopy, what you see is that the protein does not change from a beta sheet structure. There are no changes that we're seeing in the soluble species. We did a further analysis after Valeria Pierluisi-Ruiz left from her summer research. And what we found was that our preparations that were used in these experiments had a protein that had the disulfide bond and some protein that had missed the disulfide bond. So some of the proteases that may be in these human cells may be cleaving that disulfide, because these samples had been incubated at 37 degrees. We were not sure what was going on. So Valeria agreed to come to spend 1 year in our laboratory. She arrived today, and she's doing her orientation as I am giving you this talk. And one of her first experiments is going to be to modify that cysteine and also to express a cysteine mutant so that we could really tease out what is the role of these disulfide bond in the formation of amyloid fibrils. I told you I was going to talk to you about a lambda protein, and I will tell you a little bit more about it, but these results are fresh off the press. Nikita Kormshikov is a summer research fellow, undergraduate research fellow in my lab that generated this data very recently. And a lambda-6 protein also refolds reversibly. It's human-derived, and there is a scan rate dependency. And by fluorescence, he has been able to modify that cysteine. So this is an amino carboxylation in the last cysteine, or it's the second to the last cysteine in lambda. And what he's able to show is that the RCAM protein has a 3-state unfolding transition. So it seems that this modification on the C-terminus of the cysteine, it's doing something possibly to the constant domain, which is the second domain of the light chains. This is so new we are still staring at this data, but I wanted to show it to you just to tell you that these human-derived light chains are letting us do a lot of studies that we would be unable to do with the bacterial-derived proteins. And while the media, the conditions to grow them, and the constructs are slightly different, they are allowing us to produce them in ways that are, we have enough material to do biophysical studies. All right, ask the questions if you have them, because we're switching gears and there's more to come about this lambda-6 protein. We're doing well with time. All right. So some years ago, we started asking the question about, we don't have an animal model for light chain amyloidosis. In fact, for many systemic amyloidosis, we don't have animal models. There are animal models for the localized amyloidosis, especially the ones affecting the central nervous system. And while those models are not perfect, they are incredible tools. We are not as lucky, and there are many theories related to that. So we decided in the laboratory that we were going to establish a cell culture system, and Marina Organi, an incredibly talented postdoctoral fellow in my laboratory, and my collaborator, Lynn, in hematology, generated an incredible amount of data on that. So we're using a human cardiomyocyte cell line to do these experiments. And I'm summarizing all this because this is all published. So we're using soluble proteins. This is all bacterially expressed. And what we found is the following. When we incubate the cardiomyocytes with soluble protein, they are soluble when we are incubating them. What happens as they are internalized and solubilized, we are trying to understand if NIH gives us money, is that there is an increase in caspase activity, particularly with our kappa full-length light chains. Look at these increase. The kappa proteins need to generate more of a caspase activity. Remember, Will, this is a lambda-6. This is a lambda-1, and they actually do not increase the caspase activity compared to the media. These are fibrils, and these are soluble proteins, and the soluble proteins do not affect the growth of the cells. Now, fibrils do not affect the caspase activity. In fact, there seems to be less, and there's an explanation for these results, because the fibrils, particularly these kind of mustard color, and ALT05, the lambda-1, but is actually the worst in this case. It's because there is a cell growth arrest. So we have two different responses. Kappa full length is increasing caspase activity when they are soluble proteins. For fibrils, lambda-6 or lambda in general, cell growth arrest. So we started with the fibril studies. And, hold on. Yeah, and I have to go here. So these are fibrils, and we're taking a movie. Here are the fibrils. These are our human cardiomyocytes. They have red nuclei, and you may have noticed that the fibrils have a homogeneous distribution. And right now, you can see the cells are moving, and they are kind of cleaning up. All this is published in case you miss it, because I cannot show you repeat in order to allow for questions. But if you want me to show you the question in the questions, the movies, you can ask that. Okay, so summary. In this movie, the cardiomyocytes seem to be moving around to try to clear up debris from dead cells and to try to make room so that the cells that are barely growing can grow. But the result actually is that they are, oh, no, we don't want, let's go, very good. The result is, even though cells are growing, cells are dying, flatline, black in this graph. Lin, who is very interested in cell therapy, proposed to us many years ago, what about if we do a cell co-culture and we add mesenchymal stromal cells, or MSCs? And here we tested 3 donors, and we partially rescue the cardiomyocytes that were affected by the Will fibrils, these lambda 6A fibrils, from the cell growth arrest. And of course, movies show it better. So here we have, for the first day, they are growing happy. And here we're putting the fibrils together with the mesenchymal stromal cells. These cells are here. They are not labeled because they are very finicky. And you can see them behaving exactly like you're supposed to, they are supposed to behave. This one is moving really, really quickly, cleaning up. They are the garbage trucks of the co-culture, and they are allowing more of the cardiomyocytes to grow. Okay. We did RNA-seq analysis. This was bulk RNA-seq. This is a volcano plot showing you some of the genes that are, expressed more differentially, but this really doesn't tell you much. So I'm showing you here in these plots the increased expression by average RPKM. And let me show you here the boxes. These are genes that are involved in innate immune response, some complements, some interleukins, CXCLs, CSFs. These are all innate immune response. And what this is indicating is that in the presence of the amyloid fibrils, our cardiomyocytes are behaving a little bit like macrophages and are treating the fibrils as if they were a foreign agent, like a bacteria or a virus. We are also seeing changes in the extracellular matrix proteins, namely some matrix metalloproteases. Okay, there are 2 minutes. And this is the paper that we published last year in Amyloid that has a lot of data and is showing the early events of fibril formation. I'll show you just very quickly the proteins that form fibrils quicker in this analysis, the tryptophan fluorescence that changes the most. That's all I'm going to show you, guys. And if you want me to show more during the Q&A, I can go back to that. I want to summarize because I show you a lot of data. Our light chain sample altered dimer interfaces that are toxic and very dependent on somatic mutations. Our expression systems affect the behavior of these immunoglobulin light chains. The light chains expressed in human cells refold reversibly, and these light chains may refold reversibly because they have never sample aggregated states compared to our bacterial inclusion bodies in laboratories. All of our bacterial cells sample these inclusion bodies one way or another. Our soluble and fluoride light chains affect these cardiomyocyte cell line model system differently. The transcriptome is affected by the presence of amyloid fibrils, making them behave more like macrophages. And I didn't talk about this. I am the cheerleader of this incredible group of talented people. Matt Otten and I have been working very closely during the pandemic. And this is the group of people that are part of my laboratory right now. Pinaki Misra left our laboratory very recently, but he has been working very closely with Chris Dick, with Nikita Kornstikov and Valeria Pierluisi in the Human Express light chain study, the biophysics and the amyloid formation. We are hiring. We want to continue doing work in cell biology, cell toxicity. We want to do single cell RNA-seq of our studies with the cell toxicity. We have some ideas about tissues. So if you're interested, please send me an email. And I want to mention our great clinical collaborators in the Amyloid Center at Mayo, in the study of the biophysics of the Human Express light chains. Benoit Bragantini in George Muir Laboratory did analytical ultracentrifugation. The work on Will could not have been done without our collaboration with John Wall. And we have done some initial studies on solid state NMR of AL09 with Chan Ristra. We have a no-cost extension on a grant with NIH. We have been funded by NSF, Mayo Clinic, and we could not be doing this without the support of our amyloidosis patients and their families. So thank you so much. And I am ready to take any questions.
So thank you, Marina, for this very exciting talk. I'm sure that there are many questions, and I would like to ask everybody to type them into the Q&A form. So far, I think there are no questions in the Q&A.
Okay.
So this gives me the chance maybe to ask you a few things, and maybe
Let me go back to the part where I didn't, ask me, Bernd, and then whatever you want to ask, I'll move to this.
Well, the first question is obviously related to these full-length light chains, and that you said that you had this kappa 1 light chain that does not refold reversibly when you do a bacterial expression.
Yeah.
But I'm wondering now, is this due to the fact that maybe in the human cell you have any post-translational modification that changes the stability somehow? Or did you look at the protein stability produced in bacteria and in
that's
humans,
a great question.
Human cells? Is there any difference?
Yeah. So let me stop sharing and look at my slide. That's a great question. And so far from the 2 proteins that we have analyzed, and I believe we made a table of that, comparing that, the short answer is the stability is very similar between the bacterial express and the human express. Now, for the bacterial express, we have only done the scan rate dependency of a few of them. So when you don't have refolding reversibility, you can only have what we call apparent melting temperatures. You cannot do the whole thermodynamic parameter analysis. So far, because this is so new, we have done the analysis on 7 proteins, and we did his tag and on tag, as we call them, you know, without any tag in the human express. And we use the kappa 1 and AL09, Will lambda 6A, on tag and his tag. And we have started using, with AL09, we had trouble with the on tag. So we have, and we have the lambda 6 germline to compare with Will. So some of the proteins are giving us trouble with expression. They are toxic to the human cells, so they kill the cells.
Mm-hmm.
Oh, there are some questions in the chat and in the Q&A. Anyway, so I have been busy, and this paper I'm promising is going to be in bioarchives very soon. And I am hoping to have some feedback from the community because, you know, we know that the cloning artifacts may have a role. I didn't have time to show you this data, but the his tag has some effect on the fluorescence of the tryptophan fluorescence of one of our proteins. So definitely these proteins are very sensitive to their environment. And we want to do a very serious and rigorous analysis. And Chris, our lab manager, is finding plasmids that cut all of the possible cloning artifacts so that we can be able to, you know, compare as much side by side these proteins.
Okay.
I hope this answers your question.
Yes, yes. Thank you. There's one question in the chat that I would like to read for you from Tom Rothstein. Very nice talk. It appears that fibrils can be toxic to cardiomyocytes, which perhaps addresses the ongoing controversy as to whether oligomers or fibrils are toxic. Is this cellular toxicity due to fibrils outside the cell or inside the cell?
Great question. And so far, Tom, what we are seeing is that the fibrils are internalized very quickly. We have not been able to capture kind of the moment when the fibrils are on the surface of the cell. And this is more technology related rather than, you know, the cells not letting us do it. We're doing all these in live cell imaging, and live cell imaging has some restrictions, but we like the live cell imaging because the cells are alive. We're not fixing them, so which brings another can of worms. We did confocal microscopy, and so far when we are seeing the toxicity, the fibrils appear to be both on the surface and some of the protein is internalized. And again, one of the proposed experiments for our future directions hopefully will address this issue, because in this paper that we published in JBC in 2016, we know that the internalization is via micropinocytosis primarily. And then it's also, and I'm blanking, clathrin-mediated endocytosis. So there are a couple of inhibitors that we can play around with, and then we can block, and then we can see whether or not the fibrils are toxic or not. So there's so many experiments that we want to do, and not enough staff to do it. So your question is very pertinent also because in the patients, if the fibril seed is outside of the cell and this protein is in circulation, then it could actually help us determine whether or not drug targets for what is happening inside the cell are pertinent or not, or if we should concentrate on things that are happening extracellularly or not. So I hope in a couple of years to be able to more specifically answer your question, Tom.
Okay, there's another question from Martin Muschel, which goes into a similar direction. So is it really isolated fibrils versus aggregated plaques that promote tissue or cell damage?
So I'm thinking, Martin, that you are talking about more of the other associated factors that come with the fibrils. And so this would be proteoglycans, apolipoproteins, clustering, serum amyloid P, etc. So our experiments have been done with in vitro created fibrils. We have access to patient tissues, but the access that we have to these tissues is limited. And up until now, we have been saving those tissues for structural studies, similar to the work that Markus Feindrich has been doing for cryo-EM. That's what we are hoping to do. And for the type of studies that we are doing, you require a lot of material, and that's why you have to do it in vitro. It would be great, and it's a dream that we could recreate the plaque because there's this unanswered question, whether or not some of the other components of the plaque also are playing a role in the cytotoxicity. And I don't think we have an answer, and I don't think we can say that it's only the light chain, because the rest of the plaque is surrounding the light chain amyloid fibril. The fibril is kind of at the core, if you will. We don't know the mega structure of the plaque. So those experiments should be done. Now, the German group with their Forschung, I'm not saying, I don't know the entire name of the, you probably know. The German project, they may have more access to more tissues and tissue samples. And, you know, with technology advancing, if we could do these experiments in a more micro scale or nanoscale, then, or even at a cellular level, you know, one cell at a time, I think that will allow us to understand more the role of the plaque rather than in vitro fibrils. So Martin, you are on the spot, and you know, what we're learning with our in vitro cell studies could be then move forward to do cell studies with plaques. A beautiful project. And, you know, I would be happy if these German consortium of projects would be interested to, you know, share what we are doing.
Could be very interesting, yes.
Yeah.
There's one more question in the chat from somebody anonymous in the audience that relates more to the antibodies. And does light chain bind with any Fc receptors? What about the interaction between AL with immune cells such as macrophages?
Very interesting. We don't have any evidence that our light chains bind to any Fc receptors, but I don't think we have explored it to the extent that has to be explored. Now, another question kind of posed in a different direction is there is no antigen known to this disease. These cells, the cells in the bone marrow are generated through a malignancy. There's a lot of things that are wrong with this disease. And there is a study that we published years ago, and there are more clinical studies that show the chromosomal translocations and other mutations in very important genes that regulate the cell cycle and, oncogenes that talk more about the biology of the plasma cells. So, and then the other part of the question was about interactions between AL with immune cells such as macrophages. No, yes. We haven't done that. And, it is kind of interesting to be studying a protein involved in the immune cell and then the amyloid generating an immune response. It makes a little bit of your brain kind of twisting in a Gordian knot. And it also, we have to be very careful with our reagents because we use a lot of antibodies, but our proteins are antibodies. So it's kind of, it makes us run our experiments with a lot of controls, if you will. Very interesting questions though. And I think something that we should consider in the future, especially with our, if we ever move from cells to tissues or organisms.
I agree. There's one question from our organizer, from Rams. He did not actually write it into the question and answer, but he wrote it into the chat. So thanks for the excellent talk, Marina. Can you please provide molecular level insights in the lambda AL proteins, fibrils, arrest cell growth? Also, does this vary with the fibril polymorphism?
Yeah. So, something that we saw in with the RNA-seq analysis was that we believe that the fibrils are making our cells die. So in the microscopy, in the movies, you see debris with the fibrils and the cells. In the RNA-seq, what we are seeing is a phenotypic transformation. So the cells are changing their genetic makeup, if you will, and they are becoming less cardiomyocyte and they are becoming more endothelial cells, if you will. And also more macrophage. So what we are thinking is that there's probably some stress response that make them stop dividing because this stress response is generating, they're attacking this foreign agent, this bacteria-like thing that is surrounding them. They're spending a lot of their energy there. Mm-hmm. The stress responses were not as significantly changed. The p-values were not as significantly separated in the volcano plot, but they were there. So the study that we made, what we analyzed was the low-hanging fruit, the genes that kind of caught our attention. But there's so much data in these RNA-seq analysis. And now, what we're learning is that bulk RNA-seq, it could be a little bit biased. And that's why we want to do the single cell RNA-seq, but we need people in the lab to do it. And now that the pandemic, hopefully, knock on wood, is under control, most of the world, we want somebody that is really interested in coming and helping us with all of these to do that. Rams also asked about fibril polymorphism. And there, I go back to the incredible work Marcus Fendrich and Lynn Rademacher have been doing. And they have solved 3 structures of lambda light chains and then there's one structure, Stefan
Ricagno.
Yeah. Stefan Ricagno in Italy. So there are 4 cryo-EM structures from patient samples. All of them are different. All of them are different. Now, something that I want to point out is the tau protein, the same sequence. Michel Goddard has shown that from different patients, different regions of the brain, same sequence, but affecting, with different diseases, there's polymorphism. Light chains, every patient is a different sequence. We're gonna have a different level of polymorphism here. It's not surprising. The polymorphism is gonna be to an n degree. And we're just talking about, in vitro, let alone when we are talking about the plaques that come from the patient. So that's why we're saving our very precious patient samples for cryo-EM. And we are pushing Mayo Clinic to buy a cryo-EM for research because we don't own a cryo-EM. We need one because we have a lot of patient samples and we can do incredible work in the entire world because, if we know there's fibroid polymorphism for A beta, for tau, for alpha-synuclein, light chain is going to bring it to a different level. Great question, Rams. Yeah.
There's one more question from Tom Rothstein. If light chains are reverted to germline, will they aggregate? Is aggregation only a function of accidental somatic hypermutation?
Yeah. So another great question, Tom. And here I will quote the late Chris Dobson. You can make any protein aggregate under the right conditions. Now, are the right conditions what you have in the body? And first of all, the germline never has been found in the body. Our germline proteins are very hard to aggregate. The kappa-1 forms fibrils at pH 2, and it takes a while. At pH 7.4, it aggregates, but forms really very ugly, early intermediates, and it never forms fibrils at pH 7.4. So the germline seems to take a long time. In the lambda 6 germline, it's a different story. That protein has more propensity. So there are some of my colleagues, Luis Del Pozo Jauner, that is in Southern Alabama, proposes that some of the germlines may have more the propensity, and maybe that's why they are more overrepresented. And the somatic mutations just push them over the energy barrier. That is the limiting step for misfolding. And I think that's going to be the case for some of them. Kappa-1 seem to be the germline that is the least amyloidogenic. I think I answered the entire question. Yeah, okay.
So there's another question in the question and answers form. There's reports B cells can also function as APC. That's probably antibody, amyloid antibody. What about the dysfunctional B cells interaction with T cells by Murugesan Velayutiam?
Okay. APCs, amyloid precursors, is that what antigen presenting cells? Yes. Okay. Yes, they are antigen presenting cells, interactions of B cells with T cells. So in the world of amyloid, it has not been studied. And that would be a really interesting thing to study, especially in the world of COVID. Our patients are very vulnerable with COVID, and our patients make up all the patients with hematologic malignancies make up 50% of the cancer patients that have died of COVID. There's nothing that you can do with a patient with a hematologic malignancy when they contract COVID. So whether or not there are some circulating B cells or plasma cells that are interfering with the T cell interactions that are the defense mechanism against SARS-CoV-2, very interesting. Now, I am a, very basic immunologist. My immunology knowledge is extremely limited to basic B cell biology. So this is a really interesting question to pose to especially T cell biologists. And I work with a lot of them. So thank you for the question. Is there another one? Oh yeah, no, that's it. Oh, and I see Rams.
Great talk, Marina. Enjoy it very much.
May I ask one more question? I didn't type it into the question and answers, but anyways, because you have shown this diagram by Palladini about the survival rate as a function of disease, basically the after diagnosis, and I'm wondering now, is the survival dependent on the organ failure upon chemotherapy, or is it really a lack of donor organs, right? That you need a heart in order to have heart transplantation so that your heart functions, and actually would be curious to know, so where do you think biophysical approaches like what you are doing and we are also trying, what do you think, where or how can we interfere, right? When you design small molecules, which direction do you think that the field should go to
another
help
great question,
help these patients?
So if we could make our patients, the organs function better. So, and here I can make a connection with the great work that Jeff Kelly has done with TTR amyloidosis, where you can stabilize the light chain dimers and then prevent them from forming amyloid so that the organ can use its own mechanisms of, cell remodeling, tissue remodeling, and regeneration to get rid of the toxic species, being fibrils, being oligomers, being, whatever they are, both, all. Then you allow the tissue to regenerate those stem cells that reside in the tissues to then differentiate into the tissue cells instead of fighting the amyloid. Then the patient has a better chance to tolerate all the drugs that we're, pouring at them. So that is key. So giving the patients time would help them tolerate the drugs that are already in the market or the drugs that are coming in the market. That would be key. And that's what tafamidis is doing with the TTR amyloidosis. It's not reversing the disease, it's keeping the disease stable. But that is huge. That's absolutely huge. So I believe that there are some drugs in the works for light chain. And, with light chain is more heterogeneous disease. It doesn't matter if you help 10% of the patients, FDA, please approve it, because this is going to help some patients. And it could be those patients that are in this very steep curve that are dying in less than 6 months. That is important. That is helping people. But we're not going to understand how to create those molecules unless we do biophysical studies. And we understand these mutations. So.
Okay, so thanks for this great answer. There's one other question in the chat, so maybe we can have this quickly. And the question is whether the AL formation is different among the LV versus RV in the heart.
The left ventricle I believe it's the left ventricle where things, the walls of the heart thicken. But don't quote me on that because I'm not a cardiologist. This is what I have learned. So I believe that from the circulation of the heart is the left ventricle where the blood is pumped first. But you are stretching my knowledge a little bit more than I should be quoted on. But I believe it is the left ventricle where things, the wall thickening and the ejection fraction is altered. But Martha Grogan is my colleague at Mayo that has published incredible work on this. So I would refer to her publications.
Okay, so thanks a lot, Marina.
Thank you for the opportunity. This has been a great time to exchange ideas and think of new projects that we can do as a community. So thanks, Rams, for the opportunity to join
Thank you very
here.
much. Great talk, great science.
Yeah.
Thanks, Brent, for the excellent moderation. For some reason, Zoom doesn't allow us to type in the Q&A. If you're an organizer, you don't have any access to
Yeah, and you know, if some of you have more questions and you couldn't ask them or they come back, please email me. And you know, you can watch these on YouTube, I believe, and then ask me questions. I am available via email.
Thank you very much.
Thanks a lot.
Take care.
This session. Take care. Bye-bye. Bye, Ben.
Okay. Yeah.